Cancer has been introduced into all of our lives as a painful and confusing subject to discuss. However, there's a pattern to each component that causes cancer and the spread of tumors. Those cells in the corner there are our friends. They carry out their own different functions in our bodies without having to be asked. On how many occasions during the day do you consciously think of your cells? Probably close to none, of course, unless you're a STEM student. The three cells then need their own friends to help them with their tasks, and most often replicate and divide through the process of mitosis in a controlled way. Unfortunately, sometimes friends backstab, and in this case, those cells backstab us in a sense by splitting uncontrollably, causing cancers and possibly lethal tumors. Cancer-causing mutations include mutations causing uncontrolled cell division, mutations in apoptosis-inducing factors, metastasis, and angiogenesis. Uncontrolled cell division is induced by different mutations that disrupt the cell cycle regulation. The first cross-section of a layer of skin here is the first mutation, which is the precancerous state, which later develops into a benign tumor and further goes on to produce a malignant tumor, which can continue to spread. Uncontrolled division can be caused by either loss of function or gain of function mutations in certain genes. For example, let's take the p53 protein that causes cancer following loss of function mutations in its coding genes. p53 acts at different checkpoints in the cell cycle, working at the G1 and G2 checkpoints where cells can only pass if DNA is replicated, undamaged, and nutrients are sufficient. p53 is known as the guardian of the genome as it takes care of most DNA damage. Its genes are always expressed, meaning constantly being translated into its proteins in the nucleus where it's needed if DNA damage is present. If there is no damage, phosphate is not added to p53, meaning it's small enough to fit through nuclear pores where it is quickly degraded in the cytoplasm. A mutation in p53 keeps it from binding damaged DNA and prevents it from triggering apoptosis or programmed cell death of a badly damaged cell. This is a small cross-section of a tumor within normal tissue. Those cells become metastatic and enter the circulatory system by crossing the basal lamina and endothelial cells of capillaries twice, once to enter and the second to exit to form new tumors at distant sites in the body. This process is called metastasis and can happen when p53 is mutated and cells with DNA mutations divide without restriction. Metastatic cells have reduced cell-to-cell adhesion and are therefore able to leave the original site of the tumor. These are normal cell-to-cell adhesions, no additional proteins embedded in the membranes besides adherens junctions, tight junctions, and desmosomes. They also have altered interactions with the extracellular matrix. Integrins are proteins that pull out from within the cell and interact with the basal lamina of the initial membrane of a blood vessel. High amounts of a specific integrin protein allows metastatic melanoma cells to cross the basal lamina twice, as mentioned before. Angiogenesis is the development of blood vessels to feed a tumor. As the tumor grows and runs out of oxygen, it releases signals for blood vessels to grow towards it. Cancer is a difficult disease to talk about. However, the good news is there are already several forms of treatment being used. The number one way to avoid cancer and similar diseases while scientists do their science is eating healthy, being active, or in other words, giving ourselves initiatives to remain our friends.